My name is Gwendolin, and I am the lab manager at the University of Washington's Molecular Information Systems Lab. And we are really happy with lab manager, sample manager, lab key sample manager, and I'm going to tell you about some of the ways that we use it and how it's improved our research at the University of Washington. Okay, so let me give you a little background on some of the things that we do at MISL. So the Molecular Information Systems Lab started out as a collaboration between the University of Washington and Microsoft Research, and it actually came kind of serendipitously from kind of a power couple that is between these two institutions. So one of our original PIs, Luis Ceza, and his wife, Karen Strauss, are Karen's at Microsoft Research, and they decided to create an institution to study the integration of biology, synthetic biology specifically, with computer science tools. So we're in the computer science department. We're the only wet lab in the computer science department, so kind of on our own in some ways, but we are working to make computer science tools out of molecular pieces. So the most important thing to that whole endeavor is that we have really important connections between physical samples and the data that are associated with them. And sometimes that line is kind of blurred at the Molecular Information Systems Lab because we store data in samples for the most part. So having a robust connection between physical objects and the computer is very important to us. And so we were searching for a tool that made that really easy and robust connection in order to help our research. Yeah. So some challenges that brought us to Sample Manager in the first place. I started as the lab manager in August of last year. And there are a couple of things I noticed when I started. So we're a pretty new lab. I think our lab was founded in 2017. And without any other wet labs in the computer science department, we had to make a lot of decisions, kind of create things from the bottom up. There wasn't a very good pathway for us set up in our department. So there were some things that we had to figure out. In addition to things like not having central air and vacuum and fume hood and things like that, we also had to figure out our whole data management and especially our sample management system. So we had these freezers, we still have these freezers that are full of racks of tubes. There are hundreds, thousands of them, mainly tubes of DNA in Eppendorf tubes. They all look very similar. As I'm sure many of you know, it's pretty difficult to fit a lot of information onto a tube. And we had all of our information kind of spread out in a lot of different places. So as an academic lab, we have a lot of different researchers working on a lot of different projects. And there wasn't one place where people were writing down all the information about the stuff that was in our cold storage. So yeah, like I said, it's pretty hard to get a lot of information onto a tube, not enough space. And also when you're writing with Sharpie, things rub off, especially after they've been in there for a long time. We have a lot of researchers. So we have 5 PIs of the Molecular Information Systems Lab. We have about 10 postdocs, 12 graduate students, a handful of undergraduate students, and all of these types of people kind of cycle in and out on a project basis, on employment basis. It's a lot of movement. So things get lost, basically. Going through these freezers, we have boxes from researchers who have been gone for 2 years, and it's not always clear when is the right time to throw something away. I'll have people reach out to me who are like, hey, I worked at MISL 2 years ago, there's the sample that is important to my research now. I'm at Caltech. What should I do? And so it previous to having Sample Manager, those kind of requests involved me digging through every box in every freezer to look for a sample that with a handwritten label that this person might have led on it. So that all to say, Sample Manager has helped us greatly in that regard. So why do we choose Sample Manager? When I started, the previous lab manager had a plan in place for creating our own sample management system. And I came from a high throughput genomics lab in which we also had our own LIMS that we had built in-house. It was very fleshed out. It handled the ingestion of thousands of samples a day to do NGS sequencing for clinical data. And we had 3 full-time engineers working just on our sample management software at my last lab. So when I came to MISL and David, our previous lab manager, said, we're going to make our own sample management LIMS, I thought, and that's going to be my job, because we really, as you all know, it kind of takes a whole team to make something as amazing as this. So he had a plan for building out a workflow, a process, getting UW-IT involved, programming the whole thing, keeping it updated, and educating all of our users for it. And as I was, getting onboarded, all I could think was that creating this would take up all of my time. I wouldn't be able to do any other managerial things. So it's kind of looking around for some other ways that we might be able to achieve these same goals without having to make it ourselves. And thankfully, there is a great solution. Sample Manager does it all. Okay, so I did look at a good number of systems when we were choosing what we were going to do about this situation. And a lot of them were not intuitive. They were basically Excel with more buttons. One thing that I love about Sample Manager that I'll talk about is the electronic lab notebook functionality that comes with the professional subscription. A lot of these kinds of things, they would keep track of data, but had a kind of minimal or non-existent way of keeping track of the physical samples and connecting them to the data associated with them. Customizability was really important to us because there's a lot of non-standard things that happen in our lab, and we needed to have some flexibility there. And then, the ones that maybe did meet some of these criteria didn't have the best support. We would have teams across the country, across the world, not as responsive. And I really must say that the LabKey team has been fantastic. I love that they're local to the Seattle area and are always there to answer questions right away. And of course, not as pretty, which I think Sample Manager is a really gorgeous piece of software too. Okay, so some things that we love about Sample Manager. Storing all the data that you need for a sample. So I found in doing my research that, for example, just the other day, I was looking for a protein sample that someone had made. I had a link to what the actual structure of the protein was. And I was looking through an old lab notebook of someone's and trying to find this sample. And the piece of information that actually allowed me to find the sample was nothing about the sample, but simply the date that they had made it. So I was looking through all these boxes and I was trying to match 220212 among all these samples, right? And that was the only labeling that was helpful on these tubes. And once I found it, it was I knew that it had been made on that date, but I didn't really know anything else about that sample based on the information from the tube. So it took a lot of digging. With Sample Manager, all of the information that we expect our researchers to include for any given sample, all that is laid out really easily on our sample type. So we have one of our sample types in Sample Manager is a liquid MISL sample. These can be pretty flexible, but the most important thing about it for me is that it has a barcode associated with it. So we print out our own barcodes. Sample Manager does have a integration with Bartender software, but we kind of have our own solution. I print out all of these cryogenic labels that stick really well onto frozen things. Just with random numbers, I've printed out like 5,000 of them. They're just sitting in the lab. And when you're adding a sample to Sample Manager, you can just put this sticker onto your tube, and then when you scan it in, all of the information for that sample will pop up, including all of the actual times that you've used that sample. So it'll link back to your lab notebook that says, I made this sample then, I was using it for this project. And that's really fantastic for me. The auto import functionality. So like I said, we have thousands of DNA samples. A lot of those DNA samples come from the same places. So there's a few companies that make synthetic DNA. IDT is one of them. And I will say that the IDT website is not the simplest to navigate. So whenever we order a sample from IDT now, all we have to do is download the certificate of authentication, which includes a large amount of data that's associated with that sample, but the IDT database doesn't want to allow the searchability of those kinds of things. But by importing it into Sample Manager, all of that information automatically gets filled in. We can give it a home in our freezer in a very specific location, and it takes like a minute. And so there's not a lot of writing that has to happen. And now we know where all our IDT samples should live. The hierarchical freezer storage. So some of the other pieces of software I was looking at, you could kind of describe where a sample is. It's a piece of metadata. In Sample Manager, it's a lot more robust than that. So we basically have a software version of our whole lab space. We can say it's in this exact freezer, on this door, this shelf, this rack, this box, this location. And it's all stored in the software in a really intuitive kind of way. It's drop downs, and yeah, it's all hierarchical. It's really easy to move samples around. It's really easy to understand what our physical lab storage looks like just from looking on our lab key database. And it's really easy to find a sample now. So if you're looking for a plasmid that someone created 2 months ago, you can just type in the name in the search box. It'll bring up all the information associated with that, tell you exactly what they did with that sample and where they put it. Okay. Another thing that I love about Sample Manager is the lineage function. So for any sample, there's a really wonderful tool of parent-child relationships between other samples that you have in your database. So we do a lot of things where our samples kind of change form a lot. For example, here I was making custom protein. So we might design a protein based on DNA or protein sequence on a tool like Benchling. And then in the lab, it goes through a lot of steps to get that from a piece of DNA into an actual protein that we can use. So it starts out as like some pieces of synthetic DNA, then we'll have to amplify a plasmid, stick in our new region, that's a different sample. Then that plasmid has to go into some E. coli. grow those up on plates, that's a different sample, and then into liquid cultures, and that's a new sample. And then we extract that plasmid and put it into different E. coli, and then get those E. coli to make the proteins, and then finally purify out our proteins. So the whole process of creating these custom proteins have a long workflow with a lot of sample types in between. And we want to save information about all of those intermediate steps, because it's important, if the protein doesn't turn out how we thought it would, to be able to go back and find which step things might have gone wrong, especially if we're making a number of different proteins from the same kind of organisms. It helps to find out, like, oh, if all of these ones at the bottom of this tree have an issue, maybe we can isolate one step of this chain that caused it. And that's not really a simple thing when you don't have these kind of lineage trees, right? You have to rely on reading through text that might have been made by multiple people, perhaps some undergraduates who haven't been fully trained on data storage. So this makes that whole process very straightforward. And truly one of my favorite pieces of Sample Manager, which yeah, I love it, is the electronic lab notebook. So previously to getting this software, we were using Microsoft OneNote. Don't tell our Microsoft people, but it's not the greatest tool for really robust electronic lab notebook software. Sample Manager, on the other hand, has a lot of aspects to it that make it really valuable to us doing kind of this basic research sort of thing. So you can make as many notebooks as you want in the software. Entries can be broken up into, I use the entry to have like a specific like type of thing. So I have one notebook for this protein sequencing project, and I have entries for each of the proteins that I am constructing, and then I can break it up into days for all the steps that I do to make a protein. And you can add all kinds of things to these notebooks that make it really easy to replicate experiments. So maybe unlike a clinical lab, our lab, it's really crucial to be able to replicate an experiment. We want to publish papers that are verifiable by our peers. So we need to keep track, really good track of everything that we do, even while we're just exploring. So the lab notebooks in Sample Manager make that really easy. You can add a lot of different types of things to these lab notebooks. So you can attach images either in like a folder style of thing, where each entry actually has a folder where you can store arbitrary file types. You can also add images in line with the text. As you can see, there's a microscope image of a nanopore flow cell on this one. You can reference sample types. So really the thing that I find the most fantastic is having these reference abilities. Anytime that you make a sample or add something to the database, it becomes referenceable in the electronic lab notebook, such that when you mention it in your lab notebook, it creates a link to your database entry for that sample, which obviously is linked to the physical storage location of that sample in lab 2. So if you're going through trying to replicate someone else's experiment, you don't have to guess which sample they were talking about, or which set of primers that they were talking about, or plasmid, what was their starting material. You can find the literal physical object that they were handling when doing this experiment very easily, because right when they were talking about the experiment in the lab notebook, they added a reference to the physical sample with a barcode, and we can find its location. We can also add things like just general links to tools like benchling, and then the sample types, obviously you can make it however you want. So we have liquid and non-liquid samples, plates, organisms, whatever we need. So yeah, some of the things that have really improved in our lab since starting with Sample Manager. Now all of the samples that are going into our freezer have robust two-way links to the data associated with them. So I no longer have to go digging through all our freezers to find an old sample, and I don't have to dig through everyone's files to find any data that's associated with a sample. It's really easy to go both ways. All of our lab's data is centralized and accessible from anywhere. So instead of people storing files on their own computers, storing on computers in the lab, even leaving data on instruments because it's simpler, now everything is in one place and we can get to that information from anywhere just by logging in. Having it all in the cloud and online is really crucial. And I access Sample Manager from my phone, from the lab, while I'm on vacation. It's great. Another thing I love about Sample Manager is that new researchers have access to examples and information on how senior researchers conduct their work. So starting a new lab notebook, starting a new experiment, getting into research for the first time, a lot of students feel kind of overwhelmed, maybe like they don't know exactly what they should be doing, especially when it comes to recording their information. So having examples right in front of you for things like lab notebooks, having examples of what information I need to include when I'm creating a sample that I want to put into storage is really helpful for our new researchers. And I think it's great. I made a whole training how to use Sample Manager from inside of Sample Manager. So we have an example notebook that students can go into, leave comments on, ask questions. All of our slides are in there, and then you can create an entry that says when you did the training. And I just think it's fantastic. Everything is very contained, and it's really easy to find lots of good information about how research at MISL should be done and is done by the people who've been there for a while. And finally, the flexibility of the software makes it really easy to integrate new technologies, storage, and processes. So we work with a lot of different software tools, a lot of different people. We do a lot of collaborations. And all of these things feed into Sample Manager really well. IDT and AdGene, they send us physical samples and twist. Azenta does a lot of our Sanger sequencing. OneNote was kind of where we were transitioning from, and it was pretty easy to port lab notebooks created in OneNote into Sample Manager. We do our inventory with Quartzy, and Benchling is where we design a lot of our DNA and protein constructs, and all of these feed into Sample Manager super easily, thankfully. And nothing gets lost. No headaches, no researchers crying in the ante room because they can't find their sample that they made last week. We have a great workflow and a great team. So I want to thank you, Jason, again for your introduction and for hosting this conference. Thanks to Hannah and Will and John on the LabKey team for keeping us happy. And also thank you to a couple of the PIs at MISL who I work especially close with, Jeff and Luis, and Varshita and Twishi are some undergrads who helped me kind of get this set up when we started. So I'm happy to answer any questions that y'all might have. Yeah, thanks again.
Thanks so much, Gwen. I think it was it was really great to hear from you, and the story that you shared for your lab, I think, is very familiar for a lot of the folks on the call. So you're certainly in good company. So there's been a few questions that people have posted to the Q&A, so feel free to kind of add those in, and I'll be asking Gwen some of these, and then I've got a few of my own, of course. So one of the first questions, and I'm not sure if this totally aligns with the type of samples that you guys have, Gwen, but do you label the blood or tissue tubes with the same barcode and use that sample lineage option to track it from source to tissue to extracted DNA, or maybe the broader question is, are you using that the same barcode for multiple samples to link them together?
Sure. Yeah. No, we don't use the same barcode, even if we have the same source for a sample, right? So each sample in the chain is going to get its own barcode. That way we can pretty easily differentiate them. But Sample Manager provides really easy links between those samples so that we don't need to use the same barcode. So for instance, if I find a sample with a plasmid in it, it's got a barcode. I can scan that barcode, open it up in Sample Manager, and just click on its parent, which is going to be usually an E. coli that is on either a plate or it's in our deep freezer. That has a different barcode, but it also has its own location with it. So we don't lose information about the lineage by switching barcodes at each point, and everything can have a unique barcode so that they don't get confused with one another.
And then another question here, how did you handle getting your older samples into LabKey? Maybe a secondary question to that, did you relabel the old tubes, or did you just start the process with new samples?
That is a great question. So we have a lot of different sample types. Some of them were easier to get into LabKey than others. So the ones that were easiest were ones that already had some structured data associated with them somewhere. So like I said, we order a lot of synthetic DNA samples from companies like IDT or Twist. And while their databases aren't very easy to search through, they did retain information about all of those samples. So we were able to basically download the whole, database on IDT of 10,000 samples that we had ordered from them over the past few years. And the tubes themselves from IDT do have barcodes on them. They don't actually link to the samples on the IDT database, which was very frustrating. But after having IDT send us this whole very large CSV file with all of the information about all the samples that we had ordered, we were able to port that into Sample Manager fairly easily and have all of the information of all those samples become searchable automatically. And yes, we didn't have physical locations from the get-go for that, but we do have all the information that we needed about what actually the samples are. So that was really great. And then for things that were, what I would call MISL samples, ones that we created ourselves. Someone did some kind of chemistry to make the sample and then stored it in an Eppendorf tube. I kind of put the responsibility to the researchers themselves to put anything that they want to keep for the long term into LabKey and get a barcode on it. So I wouldn't call it an ultimatum, but I gave them some work. I said, anything that you want to keep in our freezers, if the box has your name on it, you need to put all of these samples into LabKey. If we don't have information in LabKey for a sample after X amount of time, then I'm going to consider those samples abandonable and get rid of them. So that put some impetus on the researchers to do a little better at not just labeling, but keeping all their information in one place. And then for things that I knew that we would want to keep, but the researchers weren't available, I got some undergrads to do that kind of thing, which is a wonderful resource.
I think that's great advice. Another question here: are you storing other reagents besides the things that are coming from IDT? And are you managing the use and knowing when to reorder reagents via Sample Manager? How are you doing that now?
Totally. So anything that comes with a barcode on it, we can kind of co-opt that barcode to use in Sample Manager. But we don't have to do that. It's easy that in our sample types, we have the ability to have like a alternative barcode and a MISL barcode on any kind of thing, right? So if we can add more information as needed. I do store reagents in Sample Manager for things that are like specific enzymes or other kinds of things that are perhaps expensive and that are used more rarely. So the thing that I like about Sample Manager is that it's easy to find something that you haven't interacted with for a while. So I wouldn't use, or I wouldn't really feel the need to put something like culture media that's in a big bottle that is in the fridge that everybody goes to all the time into Sample Manager at this point, because everyone knows where to find it. And everyone kind of like, it's something that we use all the time. But say that we have an enzyme that, it cost $500 to get half a microliter of it, and we're using it for this one project, and we know that we're going to be storing 400 microliters of it, and that someone might need that at another time. That's something that I would definitely put in the Sample Manager with a location, so that when another person comes along, maybe in a year, and they're like, I wonder if we have any user enzyme in the lab. Should I spend this $500 from my grant? They can just check Sample Manager first and find that, oh yeah, someone used this a while ago. And actually, I know how many times they thought it, and like things like that are pretty fantastic. In terms of when to reorder reagents, no, I don't use it for that at this point, but I am interested in doing that kind of thing. I've been looking in ways that we could integrate Sample Manager with some of our ordering. And I think that would be a great future endeavor for Sample Manager. And I talked to Hannah about it before too. So, and they're really receptive to any ideas that we have for improving the software. So that's really special too.
Thanks for that. And definitely, I think that is something that we want to end up doing for folks, because that centralization, I think, is really key, kind of as you mentioned before. So I'm going to ask one more question before kind of time is up here. It's going to be one from me. Do you have any advice, Gwen, for the other users today on how to gain adoption of Sample Manager within the lab?
Yeah, totally. So that's something that has been an ongoing process, I will say. Getting people to switch over from something that they're familiar with to something that they're unfamiliar with is not simple. But I think encouraging people to start new things is really great. I think making your own training goes a really long way. So I will say LabKey Sample Manager has a lot of really wonderful training materials on their own website. It was really easy for me as the manager to learn how to use the software from LabKey's training. And I think just asking the people in my lab to just go watch that training wasn't as effective as I hoped it would be. So kind of distilling it and making the training mechanism personal really has been helping a lot with getting people to adopt this work. And for other questions, I'm happy to answer them over the chat. So
Thanks so much, Gwen.
Yeah, thank you all. And thanks for having me here.